the chemistry of the carbonate system, I think, very important for us in ocean biogeochemistry. Now, okay, let's see what you think of this. If more CO2 dissolves into seawater, which of the 4 variables of the carbonate system do you think will change? And I think, again, to some of you, this is, a fairly easy answer, but for some of you, it might be a little bit more subtle to think about this. Okay, I'm getting a good response rate coming in. Keep it coming. Oh, you can of course in your answer you can select multiple choices, right? So select everything that you think will change when we have CO2 dissolving into seawater. I'll just show you the formula here again. All right, the back and forth arrows means that things can react backwards and forwards. Okay. I think most of you who have logged on have responded. Okay, so this was kind of what I was expecting, that we see quite a lot of responses for a lot of things. Now, I think virtually everybody agrees that the pH is going to change, and that's correct. The PCO2 will also change, though. Okay, if more seawater dissolves into seawater, if more CO2 dissolves into the water, then the PCO2 will have to go up, right? There's more of this dissolved CO2. Now, as CO2 dissolves into seawater, because the dissolved CO2 is part of the DIC pool, of course, the DIC concentration as a whole will change as well, right? If you put more CO2 and CO2 is part of the DIC, then the DIC increases too. But it's nice to see that quite a few of you are also thinking that the total alkalinity will change, because that is actually not the case. So the pH will change, the PCO2 will change, and the DIC will change, but the total alkalinity does not change. And this is something, this is where the chemistry underlying the carbonate system can be a little bit hard to understand when you first start working with this. Because if you look at the chemical formula here, it's of course true that the dissolved CO2 can react with water to form carbonic acid, and the carbonic acid can then break apart to give you bicarbonate ions. Okay, so if we add more CO2, we will also increase the concentration of bicarbonate ions. And I think the reason why people are selecting, why some of you responded to say that total alkalinity will go up, is of course because bicarbonate is part of the alkalinity. But the reason why the alkalinity does not change is because when you add CO2 to water and you create bicarbonate, you also create a proton. And the alkalinity is actually the difference between the carbonate and bicarbonate and the amount of protons and HSO4 and so on. So if we create a molecule of bicarbonate and a free proton, then the alkalinity hasn't changed because they cancel out, right? It's 1 minus 1 equals 0. So that means all the others change, but TA does not change. So pH, PCO2, and DIC will be affected. Now, more in general, we can think about how different biogeochemical processes will change both the alkalinity and the concentration of DIC. And this is very important for us to think about how biogeochemical processes are affecting the carbonate system. So there's a lot of information here. This is quite a busy slide, but what I'm showing you are 2 diagrams that we call the phase diagram, which are simply plotting the concentration of total alkalinity on the y-axis against the concentration of DIC on the x-axis. And TA and DIC concentration can to some extent vary independently of each other, but when one changes, or when both of these change, they affect also the pH and the PCO2. So the two diagrams in the background here, on the left diagram, the contours show you the PCO2, and on the right diagram, the contours show you the pH. And for any combination of TA and DIC, you will have an equilibrium value resulting for PCO2 and for pH. So if we form calcium carbonate, or if calcium carbonate dissolves from being solid back into seawater, the alkalinity and DIC are both affected, and the direction of change in terms of mole per mole changes is in the direction of the red arrow. Okay, so this would be if a coral reef is growing and depositing calcium carbonate, then the total alkalinity in the water will decrease and the DIC will decrease a bit. If the coral reef dissolves again, then the alkalinity is increased and the DIC is increased. Now, photosynthesis and respiration, of course, are some of the main biogeochemical processes, and those are mostly responsible for changing the DIC concentration. So if you have a lot of photosynthesis, it reduces the DIC. If you have a lot of respiration and organic matter breakdown by heterotrophic microbes, then it increases the CO2 concentration and it increases the DIC. And associated with that, and the reason for that is to do with the nitrogen cycle, associated with photosynthesis and respiration are small changes in alkalinity. And of course, if you just have direct CO2 exchange between the water and the atmosphere, then it's only representing a change in DIC, but not in alkalinity at all. So that was the example that I talked about. Okay, so I have one more question, and after that I'll not torture you with an exam any further. But I'm curious. So if let's say a coral reef is growing, or oysters are producing, growing, and all of these calcium carbonate producing organisms are producing solid calcium carbonate by taking the DIC from seawater and calcium and precipitating calcium carbonate. How is that, do you think, going to affect the source sink balance for atmospheric CO2? So if a coral reef is taking up a lot of DIC and calcium out of the water and producing a lot of calcium carbonate, is that going to be more of a sink or more of a source for CO2? I'll display my nice chemical equation again in a way that you can see a little more larger again. Yeah, I'll give you all a little more time to think about that. Okay, eight or so responses, I think, seems to be where we get to. Aha, okay. So I think clearly we can see a lot of you in the audience are quite familiar with the carbonate system already, right? So 50% of you are saying that if you take calcium carbonate out of the water, it actually leads to more CO2 emission from the ocean into the atmosphere. And it's number 2 that's the correct answer. But of course, a lot of you may be thinking about this chemical equation in the sense of, well, if you remove some of the DIC, then surely there should be more carbon uptake. But this is actually one aspect that's often misunderstood about the carbonate system. And I think it's very important to understand that the removal of calcium carbonate is not only removal of DIC, it's also a removal of alkalinity. Okay, and what I'll do, I'll quickly go back to our diagram here. You can see here when we remove calcium carbonate, it removes 2 units of alkalinity for every 1 unit of DIC. And that actually leads to the PCO2 increasing and the pH decreasing in the water. So when the PCO2 increases, it actually leads to more outgassing of CO2. Okay, so now I was asked to talk for only 20 minutes and I'm already on 23, so I'll wrap up very quickly because I wanted to just end by talking a little bit about the implications of this. And now I think this is very important for us here in tropical oceans because we, of course, have a lot of calcium carbonate production. And the example I'm showing here is from one study a few years back where they analyzed carbonate system data from a large coral reef area. And what you can see is because of this, corals photosynthesize, but they also calcify. And because of that, you can see that the PCO2 on the reef is often above atmospheric PCO2, meaning the reef is a source of CO2 to the atmosphere. And now there's a very important implication here, which is to do with the rates of ocean acidification. And now the ocean as a whole is taking up more CO2 and becoming more acidic. So that means when corals then in more acidic water also start to remove more calcium carbonate, it actually pushes the PCO2 to even higher value and the pH to even lower values. And this is illustrated in the right diagram here, which shows for data compiled from coral reefs, and I think this was a global compilation, the rate over time at which PCO2 in the coral reef waters is increasing relative to the rate at which PCO2 in the open ocean is increasing. So what this shows you is that the rate of ocean acidification in the coral reefs seems to be actually faster than the rate of ocean acidification in the open ocean. And that's basically because of a nonlinear effect in the carbonate system, where you see that if we have a low value of PCO2 and a high pH, which is what we get when the alkalinity to DIC ratio, should be, when the alkalinity to DIC ratio is high, you can see that a small change in DIC has not so much impact on PCO2 and pH, but when we've got a higher PCO2 to start with, the same quantity of DIC change, so the length of my red arrows is the same length, you can see that it leads to a much bigger change in PCO2 and in pH if we're in a more acidified condition of water. So the implication of this here is that, of course, the ocean carbon sink is driven by atmospheric PCO2. So when we've got a high alkalinity to DIC ratio, then a small increase in the PCO2 will drive quite a large change in DIC. But if we have a low alkalinity to DIC ratio, then the same rise in PCO2 will drive a much smaller increase in DIC. So what this means is that actually over time, the ocean carbon sink as a whole is expected to weaken. And that makes it very important that we collect good observational data to track exactly what the sink strength is. Okay, we can more formally actually quantify the sink strength, and that's often done with something called the uptake factor, which calculates the change in DIC for a given change in PCO2. And you can see that at a low PCO2, this uptake factor is high, meaning that for a bit of change in carbon dioxide, the ocean takes up a lot of DIC, while at a high PCO2, the uptake is much less. So this is for those of you who are very familiar with the carbonate system, this is similar to the often used Revel factor, but I think the uptake factor is a little bit more intuitive to understand. Okay, so I'll end here. In summary, the carbonate system has 4 main measurable variables, and with any 2, you can calculate the whole system. And that means that, for any balance of alkalinity and DIC, we end up with an equilibrium pH and PCO2. the ocean sink is driven by atmospheric PCO2, so that represents an increase in DIC with no change in alkalinity. So you can go back to the phase diagram and then see how that causes ocean acidification. But we can also see that biogeochemical processes greatly alter the TA and DIC, and they do that in varying proportions, and that then drives the amount of CO2 uptake or emission from the ocean. And within that, the changes are very strongly nonlinear. So at higher PCO2, the ocean carbon sink weakens and acidification rates will accelerate. So I think hopefully I've convinced you that the carbonate system is really important, and it's very important to keep measuring good carbonate system data. And I think, of course, many of us here in the Zoom are already doing that. So thank you very much, and I hope I didn't go too badly over time.
Right, thank you, Patrick. It's a really interesting talk, and also with the interactive questions. Thank you everyone as well for participating. And if you have already some question, please keep it until our discussion after the second talk, because we will have another talk from Dr. Min Soo. Yeah, she is already here. Dr. Soo, yeah, all right. Before you start, I will just read your CV, brief CV, just to get to know you better. So Dr. Soo got her PhD from Xiamen University, and also she continued with postdoctoral at Xiamen University as well, and then another postdoc in University of Southern California. And now Dr. Soo is affiliated with Hainan University of China, and she is focusing on marine nitrogen biogeochemistry and stable isotopes. So today, Dr. Soo will talk about the carbonate system and about the nutrient cycles in the ocean, nitrogen, phosphorus, and silicon. So I think we can just start to hear Dr. Soo's presentation. Dr. Soo, over to you. You have about 20 minutes for your talk.
Can you hear me?
Yes, we can hear you.
How can I share my screen?
Okay.
Sorry. Sorry, I didn't find.
You can't share your file?
Can I send my file to Faisal and?
Yes, I think Faisal can help us. Right, while waiting.
Sorry for that.
Oh, that's all right. So while waiting, if everyone has question already, you can just put on the chat box related to Patrick talk. And just to let everyone know that after this webinar, there will be a link for survey. So I hope everyone can participate in the survey. And just click the link. Later, it will be put at the chat box, and you can fill the survey. Is it Faisal?
after receiving the file?
Hi, can you hear?
Yeah, it's coming now.
Oh, okay, it's coming.
Okay. Sorry.
That's all right. We can see. We can start.
So thank you for introducing, and Professor Patrick gave an interesting talk about the carbon cycle. Then I will talk something about the nitrogen cycle. So I'm Ming Xu from Hainan University, and at first I want to appreciate for the having me today in such a fantastic forum to show my study. So I changed my title to the dinitrogen fixation and diallotrope derived nitrogen transfer in the oleuotrophic euphotic ocean. Since the nitrogen cycle is very complicated, so today I choose one process, the N2 fixation. So we know that the ocean is a carbon dioxide sink. Since 1950, the ocean has removed 26% of the total anthropogenic CO2 emissions, contribute to nearly half of the world's primary productivity, while phytoplankton. The phytoplankton is the basis of the marine food web. So the marine primary production supports marine ecosystems by utilizing carbon, nitrogen, phosphates, silicates, phosphorus, and trace metals. The primary producers, phytoplankton, responsible for carbon sequestration, so they are vital in regulating the Earth's climate. So when we dip into the ocean, we found that outside the high nutrient, low chlorophyll area, most of the ocean are nitrogen limited. We can, from the experimental data and confirming that in the oleuotrophic ocean, in most of the subtropical gyre, the primary limiting nutrient is a nitrogen. Thus, I'm curious about the nitrogen cycle in the environment from the perspective of the nitrogen. The primary production can be divided into the new production and the regenerated production. So in the oleuotrophic ocean, the new production is supported by the upwelled nitrate from the deep ocean, and the regenerated production is supported by the ammonia, which is recycled in the upper ocean. We know that another source of the new nitrogen is the N2 fixation, which fix the dinitrogen and transfer dinitrogen into the bioavailable nitrogen like ammonia and dissolved organic nitrogen into the ocean. And this bioavailable nitrogen can be consumed by other microbes like phytoplankton and bacteria. And another source of new nitrogen in the ocean is a lateral transport. In the sunlit ocean, another process, nitrification, it converts the recycled ammonia into nitrates. We treated this nitrogen as regenerated nitrogen, but not the new nitrogen. So if the nitrification occurs in the euphotic ocean, we call it this nitrogen as regenerated production, but not the new production in the euphotic ocean. So let's talk about the diallotrophs. Diallotrophs mediate the N2 fixation in the ocean and serves as a vital new nitrogen source, sustaining the primary productivity in the ocean. and contributing to the export production. The observation from the Aloha station, the observations found that the N2 fixation can contribute around 26 to 47% of export production in this area. So the ecological impact of diautotrophs on the entire allotropic existence are profound, and its implications are heavily relies on the release and transfer of the diautroph derived nitrogen. We call it DDN. So early studies have documented the release of DDN, such as the accumulation of ammonia or DON following trichodesmian blooms or in the pure culture. And these studies mainly focus on the trichodesmian. Trichodesmian is one of the diautotrophs. They quantified that nearly 50% of the N2 fixation can be released into the dissolved pool, and evidence of bacterial production enhanced, and the relatively N15 isotope data of the phytoplankton and the zooplankton also confirms that the transfer of DDN to the planktonic food web. In the field, we found that the release of DDN is partially transferred to the dissolved phase when the nitrogen fixation occurs, but limited observations concludes the dissolved fraction of N2 fixation into the data set. This is the new data set. We found that all these nitrogen fixation rates did not conclude the dissolved fraction, and the limited field data, some of the field data, they only did measurements from the surface ocean, but not the entire euphotic ocean. So the dissolved fraction profile in the euphotic zone is not well known. And in our previous study, we found that the DDN is typically in the reduced state, existing as most as DON or later as ammonia, and light can impact the DIN flow pathways in the euphotic zone. In the high light layers, the DIN can flow into the phytoplankton, while in the low light layers, reduced nitrogen can into the nitrophiles, though light may influence on the dissolved fraction of N2 fixation is also unknown. So here is a question: how DDN is incorporated vertically into the nitrogen cycle, and the role of the light in this DDN release. In this study, we utilize the N15 labeled isotope with an experimental design to accurately simultaneously quantify the particulate phase and dissolved fraction of N2 fixation rates in the Northwestern Pacific Ocean. We use the modified bubble method to increase the enrichment of the dinitrogen labeling ratio, which can signify the N15 signal. And we utilize the simulated sunlight spectrum. We put all our incubation bottles into the artificial periodical light intensity and temperature controlled incubator. And we found that the total nitrogen fixation rates exhibited a unimodal pattern with peaks at around 50 meters. And Interestingly, we found the ratio of the dissolved fraction of the N2 fixation rates to the total nitrogen fixation rates increased with depth, and the ratio expanding from 5 to 97%. And we integrated the total nitrogen fixation rates. We found that the total nitrogen fixation rates tend to have a higher ratio of the dissolved fraction. This means that the importance of accounting for the dissolved fraction in the assessment of marine nitrogen fixation rates, particularly in the regions where the relatively lower nitrogen fixation rates in the particular phase. And we found that in this study area, the UC1-B dominates in the southern and central area, while the UC1-A dominant in the north area. We found that the total NIF gene abundance and the total nitrification rates were positively correlated with light intensity at corresponding depths. This supporting high surface light intensity may suppress the total nitrogen fixation rates. This contributing to the observed unimodal of total nitrification rates distribution on the vertical scale. The ratio of dissolved phase, but not the dissolved nitrogen fixation rates, was negatively correlated with light intensity. This possibly due to the low light promoting the DDN release. So we further conducted light manipulation experiments, and we found to validate how the light influences the nitrogen fixation and the DDN release. And the results reveal that a unimodal distribution for the total nitrification rates. And we found the optimal light intensity dominant by UCYN were around 50 micromole per square per seconds. And we found that the layers exhibited the highest total nitrification rates when the in situ light intensity matches the IK. This is the maximum of the optimal light intensity that the nitrifiers like. So when the in situ light higher than the IK, the light may inhibit the nitrification rates. And when the light in situ light lower than the IK, then the light may limit the nitrification rates. So in the individual layer, the fraction of the dissolved nitrogen fixation rates exhibited a concave trend. In the low light layer conditions, we found higher ratio of dissolved nitrogen fixation rates. This indicates that the deuterotrophs may release a higher proportion of the DDN, but the DDN are ineffectively utilized by surrounding organisms, so we can observe the higher ratio. And in the higher light conditions, we also observed a higher fraction of the nitrogen fixation rates in the dissolved phase. We guess that deuterotrophs increase cellular nitrogen release to discharge the excess energy to prevent itself from the photo destruction and light inhibition of deuterotrophs and non-deuterotrophs. So our experiments in the light manipulation experiments reproduced with the downward increasing trend of the in situ observation. That's the vertical dissolved fraction ratio as light intensity attenuated. However, we found that the light manipulation experiment, the ratio of the dissolved fraction in the highlights conditions are lower than in the highlights conditions are much lower than that in the low light conditions. This is consistent with observations in the in situ stations. We've also found that the ratio of the dissolved phase of the nitrogen fixation rates in the high light layers are much slower than that in the low light layers. So we guess that the lights only cannot fully explain the high ratio observed under these in situ conditions. In our previous findings, we found that the light gradients may create niches in the upper layers with highlight conditions. The ammonium will preferentially utilized by phytoplankton, while in the low light condition, that ammonium can be utilized by the nitrophiles. So we hypothesize that in this case, in high light condition, the released DDN may assimilated by phytoplankton into particulate phase, and part of the DDN may removed from the dissolved phase. That is why we observe the low ratio of the dissolved nitrogen fixation in the upper layers. And in the low light layers, a released DDN is oxidized by nitrophiles into nitrate, and nitrate is also in the dissolved phase. So in the low light layers, we observed the high ratio of the dissolved fraction in the total nitrogen fixation rates. So to improve our hypothesis, we conducted the size fractionate experiments and to see how the DDN transfer in the highlight layers, and we found that around 15 to 39% of fixed nitrogen from the large size stereotrophs transferred to the small size biota, and we also found the DDN in the low light layers around 70% of the dissolved nitrogen fixation rates was resided in the nitrate. So this means that in the low light layers, DDN was transferred to the nitrophiles. And in our light manipulate in experiment, we also found that with light increasing, the ratio of the dissolved fraction in nitrate decreased. We know that the nitrophiles may inhibited by light. So this is confirmed that the DDN is released to the nitrophiles. So this study, the results of the detectable transfer signal suggests that efficiently release of DDN as a majority of DON enters into the relatively large ambient DON pool and is preferentially utilized by surrounding organisms like nitrophiles. So here is the take message that in this study, we utilize a modified bubble release method and the artificial periodical light intensity to collect simultaneously both the dissolved fraction and the particulate fraction in the nitrogen fixation rates. And we found that a DDN is quickly released and transferred to the surrounding biota. And we quantified that the dissolved nitrogen fixation rates in the whole euphotic ocean for the first time, and the global marine nitrogen fixation budget will enlarge 26% when we incorporating the dissolved fraction of the nitrogen fixation rates into consideration. And the direct contribution of nitrogen fixation rates to the nitrification is around 3 to 9%. And this is the first time that we proposed a two-layer structure of DDN transfer which is driven by light intensity in the euphotic ocean. And this is the story, and this story was published last year. And if you have interested, you can download it or contact me. That's my presentation today. Thanks.
All right, thank you, Dr. Su, for sharing your research. It's a really interesting experiment about the nitrogen fixation rate. So everyone, if you have any question, we will start the discussion. So if you have any question for Patrick or for Dr. Su, you can raise your hand. You can deliver your question directly in Zoom, or if you want to write your question in the chat box, please do so, and I will read the question and let the presenter answer. So I think you don't have any
Actually,
hands.
I have a
Yeah.
question for the Professor Patrick.
Yes, sure, Dr. Su.
Very interesting talk, and I think the carbonate system is very difficult for me. We know that the oyster or corals, they can calcify, and when they calcify to, but they can also release carbon dioxide. So my question is, when we consider the coral reef as a coral reef system or the oyster farming as a system, the system as a whole, the system can be treated as a carbon sink or the carbon source.
So that's a great question. I think it's still a topic for research, but I think the general consensus is that it tends to act as a whole system. That these systems tend to act more as CO2 sources. So oysters themselves, of course, they're both heterotrophic and they're producing calcium carbonate. So for oysters, it's fairly straightforward. There's two mechanisms for CO2 production. A coral reef system is, of course, a much more complex ecosystem because you've also got plenty of photosynthesis happening, both by the corals as well as also by phytoplankton. And then the organic matter that's produced can often be exported away from the reef with ocean currents. So reefs tend to be quite significant sources also of dissolved organic matter, so DOC, but also POC. But I think typically that's relatively labile forms of organic carbon that are then used up and respired by bacteria, again, quite effectively downstream from the reef. So again, I think the kind of the processes producing CO2 within the reef environment tend to dominate over the processes taking up CO2.
Yes, since the coral reef system is very is a very productive system, right?
Mm.
So, but in the daily scale, the CO2 in your presentation that may have a very higher variability. Yeah. So.
Yeah, exactly. And I think the average value for that across a day-night cycle tends to be typically more in excess of atmospheric values. So of course, coral reefs are still very important. They're more important than only to act in the carbon balance. So we should preserve them even though they're carbon, small carbon sources.
Thanks.
All right, thank you for the question and answer, Patrick. And we have questions in the chat box. I will just read it. It's for Patrick from Afdal. In your presentation earlier, it was mentioned that the formation of calcium carbonate occurs when the ocean acts as a CO2 source, thereby increasing pH. How would you explain the process of ocean acidification in tropical waters, considering that tropical waters are generally a CO2 source?
Okay, so great question. That's absolutely right. Tropical waters are often a CO2 source because it's the CO2 equilibrium is temperature sensitive. So the exact kind of rates of ocean acidification that you get vary quite a lot regionally. So in some regions which are already a strong CO2 source, they're probably not yet acidifying to a great extent if they're kind of naturally outgassing CO2. But then in other places, that outgassing can be, for example, to do with other biogeochemical processes, like the calcium carbonate formation, or organic matter inputs and organic matter remineralization. And then in those kind of scenarios, the background rate of acidification from the kind of the open ocean water, the ambient waters, will basically interact together with the additional biogeochemical processes producing CO2 in the waters. And then there's kind of a positive interaction between those two that can increase the acidification rate. So that was what kind of was suggested by some of these coral reef data that I was showing.
Right. So, Pafdal, I think if you have more questions, you can just raise your hand. And there's one more question in the chat from Camelia Tito. Hi, Professor Patrick. Thanks for the great talk. I'd like to hear your thoughts on how freshwater input affects pH levels in coastal areas with coral reefs, seagrass, and mangroves, especially since these areas have high levels of organic carbon and turbidity.
Yeah, okay. So great question. I think that illustrates how complex coastal waters are and why it's so important that we get, for many different coastal areas, good carbonate data to understand them. So fresh water itself, the salinity of seawater has some small impact on the carbonate system equilibration. But I think the kind of systems that you're talking about, where you've potentially got really large freshwater inputs that also come, with high levels of turbidity and organic matter, there, I think then often the input of the organic matter and the degradation of that organic matter becomes very important, because that acts as an additional source of CO2 into the water. So then in a setting like that, you might have very high levels of PCO2 and quite low pH. So it effectively acts like a strong acidification mechanism. So that's what we see, especially in sort of our coastal waters here around some parts of Indonesia and Singapore, where we get a lot of organic matter input. But then in other cases, and I think that's maybe a bit more like the case, for example, around Jakarta, where the fresh water is actually delivering a lot of nutrients, then you can have eutrophication. So then you've got a lot of primary production in the surface waters, but organic matter supply down into the sediments. So then in a system like that, the carbonate system can be impacted in a different way again. So I think, the bottom line is lots of variability, and it depends a bit on where you are, what sort of factors will be going on.
Right. So it really depends on the area, on the location. I hope that answers your question. Ame, if you have more questions, you can. Yes,
So
Patrick?
I have a question for Dr. Xu, and
Yeah,
I
sure.
really, as you can tell, I tend to think about carbon more than about nitrogen and nutrients, but I do have an interest in nutrients as well, and I really enjoyed your great talk on nitrogen fixation. I was really interested by these strong dissolved nitrogen release rates, and I think maybe I didn't quite understand this properly, but I think you got these very interesting trends where, especially down at depth, you end up with a higher dissolved fraction than in surface waters. And I think if I understood this correctly, you said that this is more to do with how the released DON is processed. Is this, I was wondering though, is there, with depth and therefore with light intensity, is there variation from the phytoplankton in how much of the fixed DON, how much of the fixed nitrogen they're exuding? So in terms of just the, if you have a pure phytoplankton culture, would it exude more fixed DON under low light levels than under high light levels? And could you talk a bit about that?
Thanks for your question. And actually, from the in the entire effect zone, from the high surface down to the basic of the effective layers, the light intensity with the light intensity attenuation, the total nitrification rates actually decreased with the depth. They mostly they have a peak around 50 meters, and actually in some areas of the ocean, this peak occurs at the very surface layer. But in the olitrophic ocean, they always peaks at the subsurface layer. And for the dissolved fraction, that's the release of the dissolved phase, that's we found that the fraction increased with the depth, but at the accurate the absolute nitrogen fixation rates, no matter either in the particulate phase or the dissolved phase, both of these two rates are very relatively low in the low light layers, but the ratio increased. So why you mean that you want to know how the accurate dissolved nitrogen fixation rates is this due to the diazotrope biomass, or?
Well, so I was more curious about the mechanism of the release, right? Because I always thought,
maybe there were some explanations. Maybe the virus lysis or the zooplankton sloop feeding.
Oh,
yeah.
Okay.
Yeah, we guess. So actually, in recent studies, some groups they have confirmed that zooplankton grazing, the diazotrophs may have a higher pressure from the grazing. So when the zooplankton graze the diazotrophs, they will have a high pressure of the dissolved fraction. Yeah.
Okay, that totally makes sense. Yeah, thanks.
Thanks for your question again. Yeah.
It's a really interesting discussions. And is there any more questions from the participants? I don't see any hands yet. So maybe Dr. Su, you mentioned that your method is a new method in doing the nitrogen fixation rate. And just wondering, is there any limitation on the method that you just explained in your research?
Actually, I think that for every study, the methods always have limitations. Yeah, but we just compared with the traditional gas bubble method and the dissolution method. By using the modified bubble method, we enhanced the dissolution into 96% when we inject the dinitrogen gas into our incubator, into the bottle, and with rotating, and we increase the dissolution ratio And so when we did the incubation in the same incubation time, like 24 hours, and we can have a much higher isotope signature. And then we can get the to fixation rates. Yeah.
Right. Thank you so much. So it's a really interesting experiment, I think.
Thank you.
Is there any more questions?
I guess I have a
Yes.
bit of a follow-up one, right? So you were saying that the the revised nitrogen fixation rates based on your data might be I think up to 26% higher by considering the dissolved release. If I remember correctly, I think in your measurements, this was dominated quite a lot by USIN A and USIN B. Were you
Use one from B? Yeah.
USIN
Yeah.
B? Do
Yeah.
you? Because I guess do you know whether trichodesmium and also diatom diazotroph associations might have roughly the same kind of amounts of dissolved release rates?
Actually, previous studies mainly focused on the trichodesmium, and in the pure culture in the lab or in the field study, and the ratio is around 50%. But there were still very limited studies. So if we want to know that use one A or the delta, symbiosis with delta or other groups of the deltatrophs, and we may need a lot more studies. Yeah. Thanks for. Yeah,
okay, cool.
it's a
Thanks.
cool question.
Exciting work to do for you.
Thanks.
Right. So still many more research and experiments need to do. So yeah, maybe it's also give us like, ideas to do more research in biogeochemistry, because one of the goal of this forum is to make the audience to have some research idea or maybe collaboration together. So I think everyone, if you're interested to do what Dr. Su or Patrick do, you can just contact them. And we still have a little bit more time. Is there any more questions from the participants? And I would like to remind you that we have put already the link for the survey for the feedback from all the participants and also from the speakers, of course. Please click the link and fill the questions so that we can hear we can hear your thoughts about this webinar. If we need more improvement, or if you have any ideas for the next webinar, we'll be very happy to read it. Okay, if there are no more questions. Yeah, Dr. Su, of course.
Hi, Patrick. And I just wondering, and in the future ocean, the capability of the carbon dioxide sink for the ocean, how the capability will change in the future ocean.
Yeah, well, so I think that's a really big question, right? What I presented is just the sort of very sort of simplistic picture, which is that as the PCO2 goes up and the ocean takes up more and more CO2, the efficiency with which the ocean takes up CO2 becomes lower. And that's simply because of this nonlinear behavior in the carbonate system. At a higher PCO2, the change in inorganic carbon concentration for a given amount of PCO2 change is a lot smaller. But the CO2 sink in the ocean is also a lot dependent on ocean circulation and ocean temperature. And also, for example, deep water formation rates in high latitudes. So particularly important place for the ocean CO2 sink is actually in the Southern Ocean, and it's dependent quite a lot on temperatures and wind speed characteristics and deep water formation rates. So that's of course not considered in just this very simplistic uptake factor versus PCO2 relationship that I showed. So in general, I think there's significant concerns about the fact that the ocean CO2 sink may be weakening, also because of changing patterns in ocean circulation, increases in PCO2, and ocean warming. But in detail, it's hard to know because it depends on so many different factors. So it's really a topic for both from a model projection point of view, it has to be addressed more, but also we really need to keep generating high quality ocean CO2 and carbonate system measurements to actually be able to really accurately track the ocean sink and keep quantifying the sink strength and how it's changing.
How about the CRD engineering project? Is that helpful to the
Sorry,
ocean?
say that again.
CRD, carbon dioxide removal.
Well, so
project.
That's obviously a very heavily debated question. I think I haven't worked on this topic myself much, so I don't. I have sort of some views and opinions, but I'm not primarily an expert on this topic. I think from what I can see, I think some of the ideas around alkalinity management are so trying to increase alkalinity and then thereby let the ocean take up more CO2 is perhaps, I think, some of the most perhaps one of the more maybe well understood ways in which we could do this, where we can, I think, calculate and understand the effect better, quantify the effect better, and perhaps also constrain the impacts a little bit more reliably. But I think it's a huge question, and any attempt to really remove serious quantities of carbon through CDR involves necessarily a massive manipulation of ocean chemistry. So I think whichever technique one might be considering, I find it quite a scary thought as well. So that would kind of be my response. I think it's certainly a topic that more research needs to be done on before we can simply say, let's go ahead and do this, because there's a lot of potential for negative side effects. There's also quite a lot of potential for accidentally overestimating the effectiveness of this. So, for example, if you increase alkalinity, but thereby bring about an increase in the calcium carbonate production rates, the calcium carbonate solid precipitation rates, then you're actually again removing part of the alkalinity. And that would then be offset against the alkalinity enhancement.
Thank you. Yeah. Still much more to be done.
Yes.
Exactly. Sure. Yeah. So much more to be done and questions to be answered. All right. I think it's a really interesting discussions, but I think we're reaching the end of our webinar series for today. So thank you so much, Patrick and Dr. Sue, for your great talks. It's really informative, and yeah, it's really opening some ideas and some maybe your questions as well. So we really appreciate your time and your discussion today. And I think I will just remind everyone to fill the link that already put in the chat box for feedback, and also this webinar will be available in YouTube, I think, Pak An, right? And Faisal. So for everyone, if you want to get back to relearn for what we have already discussed, you can check in our YouTube. Is there any YouTube links, maybe, Faisal? Is it IMBF YouTube channel or?
Yes, IMBF.
Yeah, so everyone, you can just search IMBF YouTube channel if you want to look back at this amazing webinar. Right, it's already in the chat box as well, the link. Thank you so much, Edward. And again, thank you, everyone. Thank you, all speakers. Thank you, Pak An, Faisal, and all the participants who actively join us in these discussions. I think we will have another webinar session next month, and stay tuned for the announcement from IMBF. And also, yeah, there's a message. Please subscribe to the YouTube, to the IMBF's YouTube channel. Right, I think I will end the webinar here. Thank you, everyone, and I'll see you again next time. Bye.
Bye.
Bye-bye.
Thank you,
Thank
Thank you
Simi.
so
Thank you,
much.
Sumin.
Thank you very much. Goodbye.
Thanks,
Yeah.
Patrick.
Thanks.
Thanks, Dr. Sue.